The Codebreaker, Jennifer Doudna, Gene Editing, and the Future of the Human Race, by Walter Isaacson. Introduction. In the spring of 2014, a scientist named Jennifer Doudna had a nightmare. She was a brilliant researcher, celebrated for a discovery so profound it was changing the very definition of life. But in her dream, she found herself being led into a room to meet someone who was intensely curious about her work. When she saw who it was, a cold dread washed over her. Sitting there, with a notepad and pen, was Adolf Hitler. He looked up at her, his face disturbingly pig-like, and said, "I want to understand the uses and implications of this amazing technology you've developed." She woke up with her heart racing, shaken to her core. The dream left her with an awful premonition. She had helped uncover one of nature's greatest secrets, a tool that could rewrite the code of life itself. It held the power to cure devastating diseases, to end generations of suffering. But her nightmare revealed the other side of that power. In the wrong hands, it could be used to design a future that was not just enhanced, but terrifyingly selective. It was a toolbox for a future Frankenstein. What was this technology that could inspire both incredible hope and such a deep, primal fear? Today, we journey into one of the most important books of our time, The Codebreaker by Walter Isaacson. This isn't just a story about science. It's a story about curiosity, competition, and the immense responsibility that comes with discovering how to play God. It's the story of CRISPR, a revolutionary gene editing tool that has given humanity control over its own evolution. Isaacson, the masterful biographer of figures like Steve Jobs and Leonardo da Vinci, turns his attention to the brilliant, driven, and deeply thoughtful scientist at the heart of this revolution, Jennifer Doudna. We'll follow her from her childhood in Hawaii, where a simple curiosity about the natural world set her on a path of discovery, to the global stage, where she found herself grappling with the moral questions that her own invention had unleashed. By the end of our time together, you will understand the fundamental secret that governs all life on Earth. You'll see how a humble defense mechanism in bacteria became the most powerful biological tool ever created. And you will be equipped to ask one of the most important questions of the 21st century. Now that we can rewrite the code of life, should we? Let's begin. Book summary. Our story begins not in a gleaming laboratory, but on the lava rock shores of Hilo, Hawaii. This is where a young Jennifer Doudna grew up, feeling like an outsider. Tall, blonde, and lanky, she didn't look like the other kids, and they made sure she knew it. She was teased and called a holy, a term for non-natives. This early sense of isolation pushed her inward, toward the world of books and, more importantly, toward the intricate world of nature that surrounded her. She found solace exploring the lush, diverse biology of the island. She was fascinated by eyeless spiders living in lava flow caves and a thorny vine called sleeping grass whose leaves curled up the moment you touched them. But she wasn't content to just observe these wonders. A deeper question burned within her. Why? What causes the leaves to close? How did that spider come to be? This relentless curiosity, this desire to understand the hidden mechanisms behind life's magic tricks, would become the driving force of her entire career. The first major clue came on a rainy Saturday afternoon when she was in the 6th grade. Her father, a literature professor, had left a used paperback on her bed. It was James Watson's famous book, The Double Helix. Doudna thought it was a detective story, and in a way, she was right. She was completely captivated by the tale of ambition, rivalry, and brilliant insight as Watson and Francis Crick raced to uncover the secret structure of DNA. But something else in that story struck her even more deeply. One of the key figures was a female scientist named Rosalind Franklin, whose critical data was used by Watson and Crick. Though Watson's portrayal of her was condescending, a young Jennifer Doudna saw something else entirely. It was a revelation. A woman could be a great scientist. More than that, the book gave her a profound insight that would shape her life's work. The shape of a molecule determines its function. Life wasn't just a mysterious force, it was a physical mechanism. The twists and folds of tiny molecules inside our cells governed everything. To understand life, you had to understand its architecture. The detective story of the double helix showed her that you could hunt for the reasons why nature worked the way it did. This realization set her on a collision course with destiny. When she told her high school guidance counselor she wanted to study chemistry in college, he just shook his head. "Girls don't do science," he said flatly, but that only strengthened her resolve. She would show him. She would do science. And she did. She went to Pomona College and then to Harvard for her PhD, and it was there that she made a pivotal choice. While the rest of the scientific world was obsessed with DNA, the star molecule of life, Doudna chose to study its less glamorous sibling, RNA. If you think of DNA as the master blueprint of life, safely stored in the nucleus of a cell, then RNA is the tireless worker who carries those instructions out into the world. DNA holds the information, but RNA does the work. It's a messenger, a builder, and, as Doudna would discover, so much more. At the time, most scientists saw RNA as a dull middle manager, just a delivery boy for DNA's commands. But Doudna's mentor, a young genius at Harvard named Jack Szostak, saw things differently. He believed RNA held the key to one of the biggest mysteries of all, the origin of life. He was fascinated by the recent discovery that some RNA molecules, called ribozymes, could act as enzymes, the proteins that spark chemical reactions. This was a game-changer. If RNA could both store information like DNA and catalyze reactions like a protein, perhaps it was the original molecule of life. This was the RNA world hypothesis, and Szostak invited Doudna to help improve it. Could an RNA molecule copy itself? For Doudna, this was a thrilling challenge. It was a chance to work on a frontier that was a little neglected but incredibly exciting. It reinforced a lesson she learned on the soccer field as a kid. Look for the niche where you can fill a role that few others can. While everyone else was racing to map the human genome, Doudna dove deep into the mysterious world of RNA. Her doctoral work was a technical tour de force, engineering a ribozyme that could stitch together a copy of itself. She was becoming a rising star in a small but profound field. To truly understand how these ribozymes worked their magic, she knew she had to see them. She needed to map their three-dimensional structure, atom by atom, just as Franklin, Watson, and Crick had done for DNA. This quest led her to the lab of Thomas Cech in Colorado, a Nobel laureate who had co-discovered ribozymes. There, in a high-risk venture that many colleagues called a fool's errand, she set out to crystallize a large RNA molecule and map its structure, a feat that had stumped scientists for 2 decades. The work was painstaking, filled with years of frustrating setbacks, but in the fall of 1995, as she was flying back and forth to Hawaii to be at the bedside of her dying father, the breakthrough finally came. An electron density map arrived from her lab that was so clear she could finally see it, the intricate, beautiful, three-dimensional shape of the RNA molecule, folded up like a piece of molecular origami. She walked into her father's bedroom and showed him the image. He was weak, but his scientific curiosity was as sharp as ever. "What does it mean?" he asked. She explained how the shape of the molecule, with a core of metal ions at its heart, allowed it to perform its chemical reactions. It was in these final conversations with the man who had first placed that detective story on her bed that she was able to clarify her own ideas. He had given her the gift of curiosity, and she was now using it to uncover one of life's deepest secrets. The structure she and her team discovered explained how RNA could be an enzyme, able to slice, splice, and replicate itself. It was the beginning of her quest to turn basic science about RNA into a tool that could change the world. By 2006, Doudna was a renowned professor at the University of California, Berkeley. Her lab was a world leader in RNA structure and a related phenomenon called RNA interference, a natural process where small RNA molecules can silence genes. She was at the top of her field. And then, she got a phone call that would change everything. The call was from a colleague at Berkeley, a microbiologist named Gillian Banfield. Banfield studied tiny organisms in extreme environments, like salty lakes and acidic mine drainage. In the DNA of these microbes, she kept finding a bizarre, repeating pattern that she didn't understand. The scientific community had recently given it a name, CRISPR. Doudna had never heard of it. She thought Banfield was saying CRISPR, like a vegetable drawer in a refrigerator. They met for tea at the Free Speech Movement Cafe on campus. Banfield spread out pages of data, her voice crackling with excitement. She explained that CRISPR stood for clustered regularly interspaced short palindromic repeats. It was a strange feature in the DNA of bacteria. Identical, repeating sequences were sprinkled between unique sequences called spacers. What was it for? Banfield had a theory, one that was just beginning to bubble up in the scientific community thanks to a quiet, obsessive Spanish scientist named Francisco Mojica. Mojica had first noticed these repeats in the 1990s while studying salt-loving microbes. For over a decade, he painstakingly tried to figure out their purpose. One day, while on a holiday he didn't want to take, he was in his lab running the spacer sequences through a database. And he found a stunning match. The spacer DNA in the bacteria was identical to the DNA of viruses that were known to attack those bacteria. It was an earth-shattering realization. He exclaimed, "Oh my goodness! The CRISPR system was an immune system for bacteria. It was a genetic library of past infections. When a virus attacked, the bacteria that survived would grab a snippet of the invader's DNA and weave it into its own genome as a new spacer. It was like taking a mugshot of the enemy. This mugshot would then be used to recognize and destroy the virus if it ever attacked again. It was an adaptive immune system, one that could learn and evolve." The beauty and elegance of this natural defense system brought Mojica to tears. Here was the answer to a battle that had been raging for billions of years, the endless war between bacteria and the viruses that hunt them. This was the story Banfield shared with Doudna. The time was right, she argued, for a biochemist and structural biologist to jump in. The microbiologists had the theory, but no one had actually isolated the molecular components of the CRISPR system to figure out how they worked. Doudna was intrigued. Here was a mysterious system that likely involved RNA, her specialty. It was a new frontier, a risky project, and that was exactly the kind of challenge she loved. She decided to jump in. And so, the stage was set for one of the great collaborations in modern science. In the spring of 2011, at a conference in Puerto Rico, Doudna was introduced to Emmanuelle Charpentier. Charpentier was a French microbiologist, a brilliant and somewhat nomadic scientist who had worked in labs all over the world. She was intense, stylish, and had a sly sense of humor. Doudna liked her instantly. Charpentier, it turned out, was also studying the CRISPR system. She had been focused on a particular type found in the bacteria that causes strep throat. This system involved a powerful DNA-cutting enzyme called Cas9, but she had also discovered a mysterious new molecule involved in the process, another piece of RNA she called tracarna, short for trans-activating CRISPR RNA. She had figured out that this tracarna was essential for preparing the mugshot RNA that guided the Cas9 enzyme. But she had a hunch it did more. To figure out the full mechanism, she needed a structural biologist, someone who could help her understand the molecular machinery at work. She needed Jennifer Doudna. As they walked along the cobblestone streets of Old San Juan, they hatched a plan. They would combine their expertise. Doudna's lab in Berkeley would work with Charpentier's lab, then in Sweden, to figure out exactly how the CRISPR-Cas9 system worked. The collaboration was a whirlwind. It was a 24-hour operation spanning continents, conducted over Skype and Dropbox. The key experiments fell to two brilliant postdocs, Martin Ginnick in Doudna's lab and Christoph Chalinsky in Charpentier's. Their goal was simple but incredibly difficult: to recreate the bacterial immune system in a test tube. For months, they struggled. They put the Cas9 enzyme in a test tube with a target piece of DNA and the guide RNA, but nothing happened. The Cas9 enzyme wouldn't cut. Then, they tried adding Charpentier's mystery molecule, the tracarna. And it worked. It was the missing piece. The tracarna, they discovered, was not just involved in preparing the guide RNA, it was essential for the final attack. It acted like a molecular scaffold, locking the guide RNA onto the Cas9 enzyme and holding the whole complex in place as it latched onto the target DNA. With the tracarna present, the Cas9 enzyme became a guided missile. slicing the viral DNA with remarkable precision. One evening, soon after the breakthrough, Doudna was at home cooking spaghetti. Watching the strands swirl in the boiling water, she thought about their discovery. She explained it to her 9-year-old son, Andy. Over billions of years, she told him, bacteria had evolved this totally weird and astonishing way to fight off viruses. And it was adaptable. Every time a new virus emerged, the bacteria learned how to recognize it and beat it back. It was a moment of double joy for her. The thrill of a fundamental discovery, and the simple pleasure of sharing that wonder with her son. But the biggest breakthrough was yet to come. As Ginnick and Doudna stood at a whiteboard in her office, sketching out the structure of the two RNA molecules, the guide RNA and the tricRNA, they had a profound realization. What if they could simplify the system? They saw that the two separate RNA molecules were intertwined. Could they fuse them together into a single, programmable piece of RNA? It was an audacious idea. They weren't just discovering something in nature anymore. They were inventing something new. They engineered a single guide RNA that combined the essential parts of both molecules. It contained the address sequence that would guide the enzyme to a specific spot on a strand of DNA, and the handle structure that would grab onto the Cas9 protein. When they tested it in the test tube, it worked perfectly. This was the true oh my god moment. They paused and looked at each other. They hadn't just figured out a bacterial immune system. They had created a programmable gene editing tool. By simply changing the address in the guide RNA, you could send the Cas9 enzyme to cut any strand of DNA, from any organism, at any location you desired. It was like a biological version of find and replace for the code of life. It was precise, it was cheap, and it was breathtakingly easy to use. They realized they had developed a means to rewrite the code of life. In June 2012, Doudna and Charpentier published their landmark paper in the journal Science. They laid out the mechanism of CRISPR-Cas9 and, crucially, described their invention of the single guide RNA. They knew it would be a new era in biology. What they didn't fully realize was that their discovery would ignite one of the fiercest scientific races of the modern era. The question that hung in the air was enormous. Their system worked beautifully in a test tube, but would it work in human cells? Answering that question became a frantic, high-stakes competition. While Doudna's lab was full of brilliant biochemists, they had little experience working with human cells. They were entering a race where other labs had a significant head start. Two main rivals emerged, both based in the biotech cauldron of Cambridge, Massachusetts. The first was George Church, a brilliant, eccentric, Wildman professor at Harvard with a flowing beard and a visionary's mind. He looked like a cross between Charles Darwin and a woolly mammoth, an animal he was actually trying to resurrect using gene editing. He was a maverick who immediately saw the power of CRISPR and, in his typically open and collaborative way, emailed Doudna to tell her he was going to try to get it to work in human cells. The second rival was far more formidable. His name was Feng Zhang, a quiet, charming, and fiercely ambitious young prodigy at the Broad Institute of MIT and Harvard. Zhang had been looking for a better gene editing tool for years. He had read the early papers on CRISPR in 2011 and had been secretly working ever since to harness it for use in human cells. When Doudna and Charpentier's paper was published, it provided him with the final, crucial pieces of the puzzle, including the full role of the tricRNA and the elegant idea of a single guide RNA. The race was on. For 6 frenzied months, these 3 labs worked in parallel, each trying to be the first to publish. It was a photo finish. In January 2013, in the very same issue of Science, the labs of Feng Zhang and George Church both published papers showing that they had successfully used CRISPR-Cas9 to edit genes inside human cells. Doudna's lab, which had to overcome a steep learning curve, published its own successful results just 3 weeks later. The line had been crossed. The tool was no longer a curiosity in a test tube. It was a technology that could alter our own species. And with that power, the dream of curing diseases was suddenly within reach, but so was Jennifer Doudna's nightmare. The world was about to learn just how quickly a powerful tool could be used, not just for good, but for glory. In November 2018, the scientific community was rocked by a bombshell announcement. A young, ambitious, and media-savvy Chinese scientist named He Jiankui declared that he had done what was once unthinkable. He had created the world's first gene-edited babies. The twin girls, given the pseudonyms Lulu and Nana, had been born a few weeks earlier. Using CRISPR, Jiankui had edited their embryos to disable a gene called CCR5. His stated goal was to make them immune to HIV, the virus their father carried. But in his rush to be first, he had broken every rule in the book. The procedure was not medically necessary. There were far simpler ways to prevent HIV transmission. The science itself was sloppy, with evidence of unintended off-target edits. And the consent he obtained from the parents was deeply flawed. The news broke just days before the Second International Summit on Human Genome Editing in Hong Kong, where Doudna was one of the organizers. The summit was thrown into chaos. Jiankui, who had planned to be a hero, suddenly found himself the villain of global science. Doudna, David Baltimore, and the other leaders scrambled to manage the crisis. In a dramatic, packed auditorium, Jiankui presented his work. He was defiant, yet seemed naively unaware of the storm he had created. He truly believed that once he succeeded, everyone would recognize his work as a great achievement. Instead, the backlash was universal and swift. Scientists around the world, including more than 100 in China, condemned his work as irresponsible and a gross violation of medical ethics. He had crossed a line that many had hoped would remain untouched for decades. He had performed germline editing, making changes to human embryos that would be passed down to all future generations. After his presentation, He Jiankui disappeared from public view. He was later sentenced by a Chinese court to 3 years in prison for illegal medical practice. The birth of the CRISPR babies was a wake-up call. It was no longer a theoretical debate. The power to engineer the human race was here. Doudna found herself at the center of this moral tempest. The very tool she had helped create had been used in a way that horrified her. For years, she and others had called for a prudent path forward, urging caution and public debate. But Jiankui had shown that a single ambitious researcher could leapfrog all the guidelines. The incident forced a global reckoning with some of the most profound questions we face. Where do we draw the line between treating disease and enhancing human traits? If we can eliminate Huntington's disease or sickle cell anemia, shouldn't we? What about making our children taller, stronger, or smarter? If these tools are only available to the wealthy, do we risk creating a genetic gap, a new class system encoded in our DNA? Doudna's ethical journey evolved. Initially, she was instinctively against any form of germline editing. But as she heard from families devastated by genetic diseases, her position softened. She came to believe that it would be immoral not to pursue gene editing to alleviate human suffering, as long as it was medically necessary and there were no good alternatives. But she remained firmly against using it for enhancements. While these ethical debates raged, the promise of CRISPR as a therapeutic tool began to be realized. In 2019, a Mississippi woman named Victoria Gray, who had suffered from debilitating sickle cell disease her entire life, became the first person in the U.S. to be treated with a CRISPR-based therapy. Doctors took her blood stem cells, used CRISPR to edit the faulty gene, and infused the corrected cells back into her body. A year later, she was thriving, free of the pain that had plagued her for decades. It was a stunning victory for science and for humanity. Similar trials showed promise in treating a form of genetic blindness and in fighting cancer. And then, in early 2020, the world changed. A new, deadly coronavirus, SARS-CoV-2, swept across the globe, bringing life to a standstill. The pandemic was a terrifying reminder of our vulnerability to viruses. But it also became a showcase for the incredible speed and power of the new life science revolution. Jennifer Doudna felt a call to arms. As her university at Berkeley shut down, she rallied her colleagues. The government was fumbling its response. It was time for scientists to step into the breach. She converted her research institute into a pop-up COVID testing lab, a feat that normally would take months, but was accomplished in days. But more profoundly, the pandemic brought the power of CRISPR full circle. The very system that bacteria used to detect and destroy viruses was now being repurposed by humans to do the same. Doudna's startup company, Mammoth Biosciences, and Feng Zhang's company, Sherlock Biosciences, engaged in another race? This time, not for patents, but to create a rapid, cheap, CRISPR-based diagnostic test for the coronavirus. By programming a guide RNA to find the genetic sequence of SARS-CoV-2, they created tests that could deliver a result in under an hour, potentially on a simple paper strip. And the story of RNA, the humble molecule that Doudna had dedicated her career to, took center stage once again. The fastest and most effective vaccines developed to fight COVID were a revolutionary new type, mRNA vaccines. Instead of injecting a weakened virus, these vaccines delivered a snippet of messenger RNA that instructed our own cells to produce a piece of the virus's spike protein. This, in turn, trained our immune system to recognize and fight off the real virus. It was a lightning-fast triumph, built on decades of basic, curiosity-driven research into how RNA works. The invention of easily reprogrammable vaccines and CRISPR-based cures meant that the evolutionary balance between humans and viruses had suddenly shifted. We may never have to face a pandemic undefended again. In October of 2020, as the world was still reeling from the pandemic, Jennifer Doudna was awakened by a 3 a.m. phone call. It was a reporter. "I wanted your comment on the Nobel," she said. "Who won?" Doudna asked, still half asleep. The reporter was incredulous. "You mean you haven't heard? You and Emmanuelle Charpentier. The Nobel Prize in Chemistry was awarded to them for their development of a method for genome editing. It was a historic moment, the first time a science Nobel had been won by two women together. It was a recognition not just of their brilliant discovery, but of the dawning of a new age, the age of our ability to rewrite the code of life." Commentary. Reading The Codebreaker feels like witnessing the birth of a new world. Walter Isaacson doesn't just give us a biography of a scientist, he gives us a front-row seat to a revolution that will shape the course of human history. There are three profound lessons that I believe this story holds for us. First, it is a powerful testament to the value of pure, curiosity-driven research. Jennifer Doudna didn't set out to invent a gene editing tool or a COVID test. She was simply a girl from Hawaii who was intensely curious about why a leaf curls when you touch it. Her entire career was driven by a desire to understand the fundamental workings of nature. She studied RNA because she found it beautiful and mysterious. That basic, unguided curiosity is what led her to CRISPR. The story of the yogurt makers, of Francisco Mojica, of all the scientists who pieced this puzzle together, reminds us that the greatest innovations often spring not from a desire for a specific application, but from the simple, joyful pursuit of knowledge for its own sake. Basic science is the seed corn of technological progress. Second, this book is a masterclass in the power of collaboration and the friction of competition. The discovery of CRISPR was a global team sport. It involved a Spanish microbiologist, French and American food scientists, a Parisian wanderer in Sweden, a Czech in California, and dozens of others. The breakthrough moment happened because Doudna and Charpentier chose to combine their strengths rather than work in isolation. Yet, the story is also filled with intense rivalries, patent battles, and races to publish that were fueled by ego and ambition. This isn't a contradiction, it's the reality of how science moves forward. Competition accelerates the pace of discovery, but collaboration is what makes the biggest leaps possible. The tension between the two is the engine of progress. Finally, and most importantly, The Codebreaker is a profound meditation on responsibility. With the power to rewrite our own genetic code, we have become the authors of our own evolutionary story. Doudna's Hitler nightmare wasn't just a dream, it was a premonition of the heavy burden that now rests on our shoulders. The story of Higianku shows us that this power is already being used, and we cannot put the genie back in the bottle. We are all now stakeholders in this conversation. It's not a discussion that can be left to scientists in their labs or ethicists in their ivory towers. It belongs to all of us. What kind of future do we want to build? What does it mean to be human in an age where our very biology is becoming a matter of choice? These are no longer questions for science fiction. They are the defining questions of our time. So, here is a simple action you can take from this. The next time you see something in the natural world that sparks your curiosity, a lizard changing color on a vine, a sunset painting the sky, don't just observe it. Pause, and ask why. Ask how it works. That simple act of engaging your curiosity is the first step on the path that Jennifer Doudna took. It's the engine that drives all discovery, and it's the starting point for understanding our place in the magnificent, intricate code of life. That was a summary of The Codebreaker by Walter Isaacson. We've explored the main currents of this incredible story today, but I'll be honest with you, a summary can only ever be a map. The real treasure is found when you make the journey yourself. The true magic of this book is in the details, the human stories, the scientific rivalries, and the quiet moments of awe that Isaacson so brilliantly captures. This summary is designed to help you decide if this expedition is for you. If anything we've discussed today has sparked your own curiosity, I encourage you to grab a copy. Reading it is more than just learning. It's an experience that will fundamentally change how you view life, science, and the future of our species. The link to the book is in the description. Also, if you found this journey valuable, please take a moment to like this video and subscribe to the Billionaire's Bookshelf. It helps us reach more curious minds like yours, and ensures you won't miss our next exploration into another book that can change your life. Let's keep learning and growing together.